Good morning everyone. So in this video we'll discuss about the ribosomes, the types of ribosomes, ribosome structure and function. Usually ribosomes were first observed by Claude microsomes at first. Later they were known as ribonucleoproteins or ribosomes. So usually these ribosomes are the protein factories. That means these ribosomes can be helpful for the synthesis of proteins. So hence they are called as protein factories. So if we talk about ribosomes, so these ribosomes are usually present in both prokaryotic cell and eukaryotic cell. I guess you might know about this prokaryotic and eukaryotic cell. So in the prokaryotic cell, there will be not well-defined nucleus, whereas in eukaryotic cells, there will be a well-defined nucleus. And prokaryotic cell usually single-celled organisms. Eukaryotes, eu means true and karyote is usually means the nucleus. Having true nucleus or well-defined nucleus is eukaryote means. These ribosomes we can find in both eukaryotic and prokaryotic cells. In prokaryotic cells, these ribosomes are scattered in the cytoplasm, whereas in eukaryotic cells, these ribosomes scatters in the cytoplasm or else it joins membrane of the endoplasmic reticulum. And some of the ribosomes can also be found inside the cell organelles like mitochondria, chloroplast. So depending upon the size and sedimentation, these ribosomes are further divided into two types. One is the 70S ribosomes, the other one is 80S ribosomes. Usually 70S ribosomes are found only in prokaryotic cells and 80S ribosomes are found in eukaryotic cells, except in mammals as they contain RBC. S usually means Swerdberg unit, that is used to measure sedimentation of the ribosomes. So the 70S ribosomes has a molecular weight of 2.5 into 106 daltons, whereas 80S ribosomes has a molecular weight of 4 into 106 daltons. So when compared to the 70S ribosomes, 80S ribosomes are larger in size. And we talk about the sedimentation or the subunits of 70S ribosomes. 70S ribosomes consist of two subunits. One is 50S, the other one is 30S. So usually these 70S ribosomes are composed of rRNA and proteins. I guess you might know about this rRNA and proteins. rRNA are usually ribosomal RNA, and proteins, as you know, that it is a chain of polynucleotides. And when we talk about 50S ribosome, these are larger in size compared to the 30S ribosomes, and it is composed of 23 srRNA, 5S rRNA, and 34 proteins. And 30S usually consists of 16 srRNA and 21 proteins. srRNA usually means small ribosomal RNA. When we talk about the 80S ribosomes, it is composed of two subunits. One is 60S, the other one is 40S. 60S ribosomes are usually larger in size when compared to the 40S ribosomes. As we have already discussed that 80S ribosomes are only found in eukaryotic cells. So the 80S ribosomes contains of rRNA and proteins as usually as in case of 70S. So the 60S, which is the subunit of 80S ribosome, it is composed of 28 srRNA, 8 5 rRNA, 5 sRNA, 40 proteins. 40S subunit has 18 srRNA and 30 proteins. So these are the types of ribosomes depending upon the size and sedimentation. So when we talk about the ribosomes in mitochondria and chloroplast, they are smaller in size when compared to that of eukaryotic ribosomes. When we talk about the structure, if we see the structure of the ribosomes, they are usually spherical in shape and the size remains constant. And as we have discussed that ribosomes are present in both prokaryotes and eukaryotic cells. Ribosomes in prokaryotic cell are smaller in size when compared to that of eukaryotic cell. So if we talk about the structure, as we said that each ribosome has two subunits. So these subunits are usually one is larger in size and the other one is smaller in size, right? So the smaller subunit usually has cap shaped structure that will be present on larger size. So the larger subunit usually has a dome shaped structure. The smaller subunit that usually carries mRNA, whereas the larger subunit carries tRNA. So the larger subunit is further has two slots. One is the P site, the other one is A site. Usually the P site consists of the polypeptide chain, and whereas A site carries tRNA that has an activated amino acid. As we have discussed that smaller subunit usually presents on the larger subunit. So these two subunits are divided by a narrow cleft. So these subunits remains freely in the cytoplasm. In the beginning of the protein synthesis, these two subunits joins together. This is due to the high concentration of the magnesium ions. So when the magnesium ion concentration reduces, then the two subunits get separated. So the separation and association of the subunit will depends upon the concentration of the magnesium ions. So if the magnesium ion concentration is high, then there will be an association of the subunits. If there is a less concentration of magnesium ions, then there will be a separation of the subunits. One more thing is that we have discussed that there will be an association of the two subunits during the protein synthesis, right? So this association of the subunits is called as dimer. Then if we talk about the function of ribosomes, so the main function of ribosome is protein synthesis, the other one is protection. So in protein synthesis, talk about protein synthesis. So in protein synthesis, as we have discussed that in the beginning of the protein synthesis, there will be the joining of the two subunits. So which is usually called as dimer. So the association of the two subunits is called as dimer. So during this process, the ribosomes attaches to the mRNA. If the more number of ribosomes attaches to the mRNA, then they are called as polyribosomes. And we have already discussed that the larger subunit consists of two slots. One is the P site, the other one is A site, right? So the P site carries tRNA that consists of a polynucleotide chain, and the A site carries activated amino acid, right? So the mRNA will be translated during this process. So the ribosomes move along the mRNA, move on to the mRNA, and as they move along the codon, the mRNA gets translated and a peptide chain is elongated where there will be an addition of the amino acids. This results in the formation of a polypeptide chain, which then further leads to the formation of protein, or this is how the protein synthesis takes place. And the second one is protection. So the ribosomes can also helpful for the protection. The mRNA that passes from the two subunits usually helps or protects the ribosomes from the nucleases and also from the proteases. I guess you all know about this nucleases and proteases, and nucleases are the enzymes that are responsible for the synthesis of nucleus, and the proteases are the enzymes that are helpful for the formation or synthesis of proteins. So this is all about the ribosomes types, structure and function.